Welcome my dear students. In today's class we will learn about one of the most important structural elements of the found in the three-dimensional structure of proteins, that is beta turns and gamma turns. These tiny loops help proteins fold compactly by allowing sharp reversals in the polypeptide chain. Let's understand their structure, amino acid preferences, hydrogen bonding and types in a simple and clear way. Let's dive in. So, what are turns in the proteins? Turns are short segments where the polypeptide chain reverses its direction. Almost one-third of the amino acids in globular proteins are present in turns or loops. Turns connect different secondary structures like alpha helices and beta pleated sheets. So what is a beta turn? It's a type of turn that connects the ends of two adjacent strands of an antiparallel beta sheet. It causes 180-degree bend in the polypeptide chain. It consists of four amino acids such as named as I plus 1, I plus 2 and I plus 3. So what kinds of hydrogen bonding is found in beta turn? The C double bond O of the first residue hydrogen bonds with the NH of the fourth amino acid residue, and the second middle residues, that is I plus 1 and I plus 2, do not participate in inter-residue hydrogen bonding. Amino acids which are common in beta turns: glycine, basically it is very small and flexible, so allows sharp bending, and proline, often found because proline can adapt cis configuration which is ideal for tight turns. Common types of beta turns: there are several types of beta turns, like type 1 and type 2 are the most common. This is the type 1 beta turn and this is type 2 beta turn image. Type 1 beta turn occurs more than twice as often as type 2, and type 2 beta turn always have glycine as the third residue because it is flexible and fits well in tight spaces. So, what hydrogen bonding in beta turns? A characteristic hydrogen bonding forms between the C double bond O of the first residue and the NH group of the fourth residue, and these hydrogen bond help stabilize the beta turn. This is the type 1 beta turn and this is type 2 beta turn. Location in protein: beta turns are usually found on the surface of proteins because the middle residues can easily hydrogen bond with water. Trans versus cis peptide bonds: in most amino acids, more than 99.95% of peptide bonds are trans, which are very stable in nature, but for peptide bonds involving proline, about 6% are in the cis configuration, and this is much higher compared to other residues. And the cis form is very useful in creating tightly packed beta turns. Another less common turn is gamma turn. It's a three residue turn: I plus 1 and I plus 2. It has a hydrogen bond between the first and third residues, and it is much less common than the beta turn. So, to sum up, the beta turns and gamma turns, which play a crucial role in giving proteins their compact three-dimensional architecture, from glycine and proline residues to the unique hydrogen bonding patterns, these turns help proteins achieve stability and proper folding. Understanding them will strengthen your concepts of protein secondary structure and help in exams like UG, PG and NET. Hope this gives you a clear picture. Stay curious and keep exploring biology. See you in the next class. Thank you.